For the last talk of the day, we will revisit the exciting question of capturing adaptive diversity or genetic diversity for in situ conservation, and Clare TRIVEDI will give the talk.
Thank you. So yeah, my talk is also another very applied talk that is looking at the UK National Tree Seed Project. I thought it would be useful to present this project as a case study of how we've captured genetic diversity in ex situ collections and how we've worked with geneticists. So two of my co-authors, I'm not a geneticist, but two of my co-authors are, and I'm lucky enough that Sean has already introduced his work, and I'm going to give a little more detail on a project he did with us as well. Okay, so I'll start by introducing the UK National Tree Seed Project, outline to you what it is. Then I'm going to outline the genetic questions that the project threw up for us and how we went about answering those. Then I'll briefly look at how we evaluated our success, and then I'll outline how the project helped to facilitate development of a UK strategy for forest genetic resources. So just to give you a little bit of context, the Millennium Seed Bank is part of the Royal Botanic Gardens Kew in the UK. It's the largest seed bank in the world in terms of the number of species which it holds. So it's all focused on native species, over 38,000 species conserved at present. And it's also a global centre for research and training in seed conservation. All of our projects have two kind of twin aims. So the first is obviously long-term conservation of genetic diversity in order to safeguard it against the threats in situ. But also we have a very immediate benefits of developing very accessible and well-documented collections which are available for research right now. So alongside the global programme, we've got a really well-established and active UK programme that's already conserved at least one sample of almost all species in the UK flora. But we really recognise that that's not good enough, and if these collections are going to be really useful for conservation and for research and for restoration, they need to have captured the genetic diversity of each species, and so that's what we're particularly trying to focus on now. And that was the explicit goal from the beginning for the Tree Seed project. So as its name implies, it's obviously focused on UK forest genetic resources. It aims to provide a national collection of both the seed itself, but all the associated knowledge that comes with that. And as I said, with twin aims of both providing long-term conservation, but also to make resources available to better understand and manage our trees and shrubs. So the first output of the project is around making those genetically representative collections, and we've done that working with over 30 different organisations, local and national, governmental and non-governmental organisations. But because we recognised from the beginning we weren't going to be able to meet that aim without both identifying and then overcoming constraints, we included a research element from the beginning. Crucially, so this picture may not demonstrate this, but we didn't want to send collectors off into the countryside without knowing exactly what they're collecting, where they're collecting it from, and how. So obviously the starting point for developing our sampling strategy was existing guidelines, and Sean's already spoken a little bit about these. Very many guidelines exist, including many that have been written by the Millennium Seed Bank. But they tend to take the collector through these three levels of questioning in terms of how to capture genetic diversity. So how many populations do you need to collect from per species across the range? How many individuals from each population? And then how many seed from each tree, in our case? And as Sean's explained, the guidelines, and based on that Marshall and Brown work, have given a very clear indication of the answer to that middle question, and it's become very established that you collect from 50 individuals per population, whilst the other two questions are a little more open-ended. When we're talking about collecting the genetic diversity, as has been mentioned throughout the last couple of days, we want to collect all of the diversity present. And it's easy to collect the common alleles because they're going to be available wherever you collect from. What's harder is to collect these other three kinds, and we particularly want to focus on those alleles which are common at the local level and are likely to come from local adaptation. So our starting point in this case was to do a desk-based study, and this was based on a method that had been developed by our colleagues at Royal Botanic Gardens, Edinburgh. It basically just looks at the literature to see what is known about the genetics of each species, what's known about the population structure, what's known about the distribution, and what's known about the biodiversity. And we did this for 44 different species. These are available. They are on the Kew website, but they're very hard to find. So give me a shout if you might want to look at those. They didn't actually help us that much because, as expected, they showed us we didn't really know enough to develop individual sampling strategies for each species, but that for most, it was likely that the populations were fairly similar across the country. So we then adopted the seed zone approach that had already been put in place by our Forestry Commission, and we suggested that we will treat a species as fully represented when it's being collected from every seed zone in which it occurs, and that includes altitudinal seed zones above and below 300 meters as well. And in that way, we really hope that we'd collect those alleles which are locally adaptive or rare, because we're collecting across both geographic and ecological space. But it was a bit of a proxy, it was a bit of a hope. So obviously we mapped the distributions of each species, we superimposed these onto the seed zones, and we got this horrible matrix which you can't read, but each column is a species and each row is a seed zone, and so all the coloured dots are our target collections that we wanted to make. Okay, so that was how we set about working out how many populations to collect from across the country. That didn't answer how we sample within populations, and population was a really difficult word for us because UK tree populations are very, very fragmented. So you end up with tiny woodlands dispersed, and it's hard to know whether the population is actually the whole country or one woodland or a group of woodlands. It's really hard to know. We also got really hung up on this 50 individual rule that is in all the seed collecting guides. We found that a team of 4 tree seed collectors could only collect from a maximum of 15 trees in one day, and obviously we had limited collecting resource, as most people do. We also had the issue, because the woodlands are so fragmented, you very rarely, or if ever, find 50 individuals in one woodland. So we ended up asking collectors to make sure they collected at least 15 individuals from across a seed zone. Within a woodland, we encouraged them not to collect from related trees by collecting trees at more than 50 meters apart, to collect from right across the canopy of each tree, and to collect from at least 10,000 seeds to make collections that are going to be big enough to be useful. Every mother tree was tagged and georeferenced, and all that seed is banked separately. And crucially, we didn't select for any traits whatsoever. So we did collect a lot of seeds. The first phase of the project ran until 2018. We collected over 10 million seeds across 60 species. We also collected a lot of field data, germination data, processing data. Every collection is X-rayed and has a herbarium record. And I should emphasize that all of these collections are available for use if any of you are interested in doing so. But have we captured the genetic diversity of the flora? So we commissioned 2 studies, the first of which Sean carried out for us. And we focused it on ash because, as you've just heard, ash is severely threatened. It's a very common species for the UK, but it's under significant threat. So in the first years of the project, we really focused on making sure we'd captured that ash diversity before it was gone. And we asked Sean, has our project adequately captured the genetic diversity, given that we've been to 59 sites, collected from 659 trees and over 2 million seed, or should we have done our sampling different? Sean built a model based on distribution data and a few genetic studies that do already exist to predict the existing distribution of diversity across the UK populations. His model then sampled from that modelled population in a variety of different ways, including the way we actually had done it, and then compared what had been captured in those sampling approaches to the total. And thankfully for us, he found that we do appear to have collected a good proportion of the diversity. So if you look at all alleles, over 90%, but even better, if you look at those locally common alleles, we should have captured 96%. That's based on capturing 1 copy of each allele. As Sean was saying earlier, you might want to capture more than that, and so the smaller figures in brackets are the percentages if you look at 50 copies of each allele. Overall, again, he found that random sampling was best, but that the approach we'd used, that seed zone approach, had performed nearly as well and far better than any other approach we might have used. The model also looked at diminishing returns, how much genetic variation might be gained if we added additional populations or trees or seeds to the sampling effort. And he finds, in short, that it's always better to collect from more populations. That isn't shown on this graph, but the population graph flattens out much more slowly. It's always better to collect from more trees than from more seeds. And that for this particular model, diminishing returns were observed after 30 populations, around 20 to 30 trees and 10 to 15 seeds. But the really interesting thing for us was this concept that Sean already introduced earlier of genetically equivalent sampling strategies. So you've already seen this figure. It gave us comfort that to collect from 45 populations with only 2 trees per population is just as good as collecting very many more trees, that 50 trees per population, but from less populations. And that model worked for us because we had lots of volunteer collectors right across the countryside. So I'm going to whiz through. The second study was carried out by Roberta, who is a geneticist at Kew. She did those initial desk studies, and she'd picked out yew as a species that really needed more work. So she compared using experimental empirical studies. She used microsatellites to investigate the diversity in 13 wild populations, and then compared that to the diversity that had been collected in 12 seed collections. You can see, again, there's a lot of gene flow. There's only 2 main gene groups, and that means the populations are quite similar across the country. The figure on the top left shows in red the diversity that had been captured in the seed collections, and in blue the diversity that had been captured in the wild populations. And her estimate is that we've captured 86%. So again, it seems to be showing that the sampling strategy has worked. And similarly, the bottom right graph, this shows the allelic capture in the different wild populations, and it shows that the lighter dots and triangles are the ones where we sampled from more trees. So it confirms, again, it's better to collect from more trees than to collect more seed from the same number of trees. So really quickly, if you don't mind, just thought it would be useful to highlight that as we were doing this work, we were working with all kinds of people. We were working with geneticists, we were working with tree breeders, we were working with people who were looking to develop seed orchards and seed stands. And that perspective allowed us to see the need to bring together that work more formally within the UK. And so I've been lucky enough to work with some great co-authors, several of whom are here today, to bring together a strategy for UK forest genetic resources. And it is based on this fact that in situ conservation, ex situ conservation and research are all tightly mutually supportive activities, but that we need to share the outputs of those work and make our collections and our data more accessible and work together in a more coordinated way to be more effective. And that's recognised in the 3 objectives for the new strategy. Just to give you a very brief update, the action plan for the strategy is now under development. Stephen was telling me last night we've already designated formally the first GCU in the UK, which is really exciting news. And we've got more to be added to the UFGIS database very, very soon. And we're looking at how to make all of our collections data more accessible in one place. So just to thank all my co-authors and also to thank UFGIN and INRA for all of their support developing the strategy for UK FGR.
Are there any questions for Clare?
I'm sorry if I missed the information, but you mentioned that you sample 15 trees per population and 10,000 seeds. So how many seeds per tree? For instance, if you have mother trees which are more proliferate and they have more seeds, do you sample more from them or not?
So that the 15 trees was our guideline. So that wasn't what we always got. Sometimes we did manage to collect from more trees, and sometimes we managed to collect from less. And similarly, the 10,000 seed to collect from was also our aspiration, and sometimes we got more and sometimes we got less. So.
Yeah, my main question is, was the representation of each mother tree in the seed crop equal or not?
Was it?
If the representation of each mother tree to the seed crop, if it was equal?
Equal. Yeah, so we would collect, not necessarily, we might collect more from a tree that had more seed on, but we would always spend more time going to more trees if we could. Does that make sense?
Yeah, it makes sense practically, but
in
actually,
the field, you have to obviously be practical, and
actually,
it depends.
It should probably be considered that you might have a bottleneck from the maternal part. For instance, if you select for few mother trees, despite the factor that you have many pollen contributors, there is a bottleneck in the number of mothers. So the genetic variation you are collecting is reduced compared to the ones you should have if the contribution of each mother was fairly equal.
Yeah. No, we do recognise, and that's all in our database, exactly how many seed we have per tree. So you can look at that variation. Yeah.
Mark Villa from NRA. I have a question concerning black poplar. So it's where seeds are difficult to store on the long run, and also in the UK there are very few female black poplar remaining. So what is the situation on this species?
Yeah, so that could be a whole other presentation in itself. We have done a whole study on black poplar. So we hand-crossed black poplar from the 2 sites where there are enough females and males coexisting, and we did manage to harvest seed that has been successfully stored. We've then taken those progeny and we've grown them on in the glass house. And Roberta has also looked at the genotypes of those. So hopefully we have actually created some additional new genotypes that's stored in the seed bank.
Devrim from Freiburg. Maybe more of a comment. You mentioned that more than 50 meters, I think you consider to prevent relatedness. But as we all know that this changes, these dispersal distances and capacities can change from species to species, particularly for excelsior. It could be like 100 meters, majority of the pollinations, for example, and seed dispersals. So in this case, perhaps the second option of sampling strategy, seed collection strategy, where you sample more populations but less individuals in the case of high fragmentation in the UK, in the forests. Perhaps that's a better strategy for the species with high dispersal capacity.
So for every species, we did collect from very many populations across all the seed zones. You'll have got a flavour from this project. It's a very pragmatic project. It was very, a lot of having to make decisions on the hoof in the field. And essentially, the guidelines we gave were to try and maximise the genetic diversity captured. But in any given woodland, we just had to try and do the best we could. And the point is that the collections are now there, and those kind of studies can be done to look at this.
Yes, but it's quite difficult.
Yeah, it's really difficult.
Yeah.
So at the end of this session, thank you very much for attending. A big applause for all the speakers.